So good morning, good afternoon, and good evening, everyone. Thank you, Ryan, for the introduction, and thank you, everyone, for taking the time and tuning in to today's presentation, where I will discuss how model-informed drug development and pharmacometrics, in specific for this presentation, can support post-approval activities, which include fulfillment of post-approval requirements, label extensions, and lifecycle management. So just a quick copyright reminder. Here I'm showing you an outline of the typical drug development process. And for today's talk, I will be focusing on the post-approval stage and mainly how pharmacometrics analysis can support the post-approval stage. So you've got your drug approved. Congratulations. But what now? You have a label with an approved indication and you have an approved dose for that indication. But you may have gotten post-marketing requirements or made post-marketing commitments, which you will need to fulfill. Or you may intend to change the dosage form. You may have to investigate further drug-drug interactions, or you may want to extend your label to special populations like pediatrics, the pregnancy, or the elderly. So let's take a look at how these areas can be supported by utilization of model-informed drug development. In a fairly recent publication from 2019, the value proposition of model-informed drug development was discussed, and that its acknowledgement in the PDUFA 6 authorization provides an excellent opportunity for industry and regulators to collaborate in further advancing the application of MIDD and thus potentially changing drug development paradigms. Today, I would like to focus on some of the areas where MIDD, when applied, can be supportive of approval by alleviating clinical trials, for example, in pediatrics and special populations, or supporting the exploration of new opportunities. So let's talk a bit about the pediatric dosing challenge. Pediatric patients should have access to drugs that have been thoroughly and appropriately evaluated, which means they are safe and effective. But one of the biggest questions to answer is what dose should be used? So in a review from 2015, which looked at pediatric trials submitted to FDA from 2007 to 2014, it was shown that 42% of the submitted trials ended in failure to show either sufficient safety or efficacy. And interestingly, 10 of the failed trials due to efficacy reasons cited dosing as a contributing factor. In a publication from 2019, the time from adult labeling to pediatric labeling is quoted at around 9 years. Now, other publications put it at 6 to 8 years, but in any case, there is a significant time lag. And this shows that there is a big need for improvement to bring drugs to the market in pediatrics faster without compromising efficacy or safety. And that's where MIDD offers three main advantages: the ability to effectively select an optimal dose or dose regimen, the supporting evidence which can be gained to effectively bridge knowledge gaps and support decision-making, and the ability to inform and optimize trial design. So let's take a look at examples where MIDD was successfully applied. I will give some quick background on Zerbaxa, which is a combination of ceftolozane, a cephalosporin antibacterial, and tazobactam, a beta-lactamase inhibitor. It is approved for treating complicated urinary tract infections and complicated intra-abdominal infections in adults at doses of 1.5 gram. That is 1 gram of ceftolozane and 0.5 gram of tazobactam in patients older than 18 years old with creatinine clearance greater than 50 milliliter per minute. Zerbaxa has not been approved in pediatric patients. So section 8.4 of the current label just simply states safety and effectiveness in pediatric patients have not been established. So the overall goal of the utilization of MIDD is to fill that section of the label as fast, as efficient, and as safe as possible. Hence, to support dose selection, Hence, to support the dose selection for a planned phase 2 study, simulations of various dosing regimens in pediatric patients were conducted to assess each regimen's plasma exposure and the probability of pharmacokinetic and pharmacodynamic target attainment. So if you're interested in a little bit more details than what I'm presenting today, my colleague Yogesh Patel gave an excellent overview of this model in yesterday's presentation during the phase 1 track, which, in case you missed it, will be available later on our website under resources. So for the Zerbaxa phase 2 dose selection, prior knowledge coming from the adult population PK model was combined with the observed data obtained from the pediatric phase 1 study to build a pediatric population PK model, which then was used to simulate exposures and probability of PK/PD target attainment to ultimately select the dose for the phase 2 study. So here we show you, to orient a little bit your eyes on what we look here is on the x-axis, you see the minimum inhibitory concentration for ceftolozane on the left and the concentration for tazobactam on the right. And on the y-axis, we see here the probability of target attainment. And so based on the pediatric simulation results, the Zerbaxa dosing regimen that best met the pre-specified PK criterion were the adult regimen of 1 gram ceftolozane and 0.5 gram of tazobactam for patients 12 to 18 years old, shown here as the green curves with plus symbols, and 20 milligram per kilogram ceftolozane and 10 milligram per kilogram tazobactam for patients from birth to 12 years old. And in terms of the pre-specified PK/PD criterion, these dosing regimens achieved a probability of 90% target attainment for a minimum inhibitory concentration of 4 microgram per milliliter for ceftolozane, here shown on the left, and 1 microgram per milliliter tazobactam, shown here on the right. So similarly to the previous example, potential fremanezumab doses for pediatric patients with migraine were evaluated using MIDD, combining prior knowledge from adults with data collected in a phase 1 study in pediatric patients. Fremanezumab is a calcitonin gene-related peptide antagonist indicated for the preventive treatment of migraine in adults. 2 subcutaneous dosing options for fremanezumab are available to administer the recommended dosage of 225 milligram monthly or 675 milligram every 3 months. And here again, section 8.4 of the label is empty, and the goal is to fill that section of the label with a safe and effective dosing recommendation. So very similar to the previous example, I outlined here that prior knowledge coming from the adult population PK model was first used to predict the phase 1 dose, and then combined with the observed data obtained from the pediatric phase 1 study to build a pediatric population PK model, which then was used to simulate exposures and ultimately select the dose for the phase 3 studies. So again, to orient your eyes, we see here on the x-axis various simulated doses ranging from 60 all the way up to the adult dose of 225 milligram. And on the y-axis, I have shown here the AUC from 0 to 28 days, so over the whole dosing interval. So based on the simulated data and exposure matching with the approved effective adult dose of 225 milligrams subcutaneous monthly, fremanezumab 120 milligram in pediatric patients weighing less than 45 kilogram was supported. And the utilization of modeling and simulation also supported that no additional body weight cutoff below 45 kilogram is warranted, since it was shown that the exposure ranges are acceptable in the lower weight group. And similar results to what I have shown here was also shown for the exposure measures like trough concentration and maximum concentrations. Next, let's talk a little bit about the pregnancy dose selection challenge. So pregnancy is a very complex state where changes in the maternal physiology have evolved to favor the development and growth of the placenta and the fetus. So these adaptations may affect pre-existing disease or result in pregnancy-specific disorders. Similarly, variations in physiology may alter the pharmacokinetics or pharmacodynamics that determines drug dosing and effect. One example here is the increased CYP3A activity in pregnant women. So model-based approaches were shown to support dosing recommendations in pregnant women. And the example I'm outlining here is that based on MIDD, it was determined that there is no need for dose adjustment during pregnancy for lopinavir, which is an antiretroviral drug to treat HIV. So lopinavir is indicated for the treatment of HIV in infected adults and children of 14 days of age or older. And so this label actually has information under section 8.4, but in 2015, at the time of the work I'm outlining here, it had no dose recommendation under section 8.1, which is the use in specific populations, namely the pregnancy. So what information was used to determine that there is no need for dose adjustment during pregnancy? 6 clinical studies of HIV-infected adults were included in the analysis, 3 of which were with non-pregnant subjects, and 3 of which were conducted with pregnant women. The concentration time data were pooled across all studies and analyzed using a population analysis approach. To account for increased CYP3A4 activity in pregnant women, a clearance increase of 17% was estimated for 2nd and 3rd trimester, while a drop of 26% in clearance postpartum was estimated when compared to non-pregnant subjects. Additionally, the derived exposures were then used to compare between non-pregnant and pregnant subjects, as well as compare the HIV RNA response. So shown here, we have on the x-axis the exposures from the non-pregnant subjects all the way to the left, stratified by formulation. And we show here also the tablet and soft gelatin capsule formulation, the soft capsule formulation shown here as the gray box, as well as exposures across the 2nd and 3rd trimester of pregnancy for standard doses of 400 and for the increased dose of 600 milligram. And despite the increase in clearance during the pregnancy, it was shown that pregnant women still maintain lopinavir exposures that is similar to the efficacious exposure in non-pregnant subjects for both AUC and for the pre-dose concentrations for the standard dose of 400 milligram lopinavir. And furthermore, substantial reductions in HIV RNA were achieved by the 3rd trimester for all doses of lopinavir administered, and no correlation between the virologic response and lopinavir exposures was apparent. So shown here in this figure is the virologic response as measured in HIV RNA copies per milliliter on the y-axis versus lopinavir AUC and pre-dose concentration values in the 3rd trimester of pregnancy. And in this range of observed exposures, there does not appear to be a relationship between the observed exposure and HIV RNA levels. And when we look at it now from a percent responders perspective, meaning less than 50 copies per milliliter, we do see that there was a lower response rate observed in pregnant women in the lowest quartile. Oh, sorry about that. In the lowest quartile. However, these response rates were similar between this quartile and those of the non-pregnant adults across all quartiles. So based on these pharmacokinetic and pharmacodynamic evaluations, it was determined that lopinavir tablets and doses of 400, 100 milligram lopinavir ritonavir twice daily during pregnancy is appropriate, and no dosage adjustment for lopinavir is recommended. So ultimately, by utilization of MIDD, a label change was achieved to include the recommendation for pregnancy. In summary, I hope that I have illustrated so far the significant value which modeling and simulation can add during pediatric drug development, specifically in selecting and optimizing the pediatric dose in special populations like during pregnancy, or another example would be the elderly. And furthermore, model-based approaches have been shown to leverage existing knowledge to ultimately bridge knowledge gaps. So similar to what I have shown how MIDD can be utilized to support dose recommendations in special populations, MIDD can support justification of dose and dosing regimen in different geographic populations, and hence ensure bridging to other markets and support a drug's lifecycle management. An example I want to use is quizartinib. The overall goal was to support justification of quizartinib dose and dosing regimen in Japanese acute myeloid leukemia patients. And with the help of MIDD, the conclusion was that the same dosing regimen can be used in Japanese and non-Japanese AML patients. So let's take a look at how this was accomplished. 7 non-Japanese studies were conducted and 2 Japanese studies in AML patients. All this information was pooled together and a population PK model was developed. During these analyses, it was shown that the Japanese population exhibited similar PK as the non-Japanese population, and hence no covariate related to the geographic origin was included on the PK of quizartinib and its metabolite. And based on the developed population PK model, exposures from Japanese and non-Japanese subjects were then compared, and fact of So in conclusion, here we see that similar PK profiles of quizartinib and its metabolite were observed in Japanese and Japanese AML, non-Japanese AML patients. And ultimately, these results supported the same dosing regimen for quizartinib in Japanese and non-Japanese AML patients. Now, what about label extensions, for example, to add additional drug-drug interaction information? In this case, for example, the development of a physiological-based PK model can support guidance of dose adjustments for co-administered medications. At this point, I want to mention that the efavirenz example, where a PBPK model was developed and validated to evaluate the impact of moderate CYP3A4 induction on CYP3A4 metabolized compounds. And if you are interested, you can stop by at the poster presentation of my colleague Inga Darling, who presents this work this afternoon. So when we think about life cycle management of a drug, often we also consider new indications. That is where the development of an exposure response model can be used in predictions to other indications and hence support approval there. So one example I want to show today is abatacept. Abatacept was approved for the treatment of adults with moderately to severely active rheumatoid arthritis. And the goal of the analysis I'm showing was to support the regulatory submission and approval process to determine whether the proposed intravenous and subcutaneous dosing regimens provided near maximal efficacy and were therapeutically equivalent in patients with psoriatic arthritis. In this example, the relationship between abatacept exposure and the probability of ACR20, which is a measurement for the improvement of disease, was modeled via a maximum effect type relationship. And on the y-axis here, we see the probability of ACR20 versus on the x-axis, the minimum concentration at steady state. And you can see here that both the 10 milligram per kilogram intravenous dosing, as well as the 125 milligram subcutaneous dosing, achieved similar probability of ACR20. And similarly, when translating this to the probability of ACR20 on day 169, you see here that it was shown that both the 10 milligram per kilogram intravenous dosing, as well as the 125 milligram subcutaneous dosing, would achieve comparable probability of ACR20 in psoriatic arthritis. And this led then to the conclusion that both formulations and their associated dosing regimens are deemed to be therapeutically equivalent for the treatment of psoriatic arthritis. So in conclusion, in the context of pharmacometrics in post-approval, multiple model-based methods may be considered. I have shown within the context of model-based approaches in pediatric drug development that exposure matching can be applied. And in the context of dose selection for pregnancy, a combination of exposure matching and extrapolation of efficacy can be applied. When warranted, more mechanistic approaches like physiological-based pharmacokinetic models may be considered, as may be done in the context of pregnancy or when considering drug-drug interactions. And lastly, Bayesian techniques to leverage prior knowledge are frequently invoked within population-based modeling frameworks, since many modeling approaches include options to inform predictions based on prior knowledge, data, or both combined. And the examples shown today, of course, only represent a small sample size of MIDD in post-approval, but I hope this gave you a flavor of what can be supported via MIDD to basically unleash its full potential and to maximize its impact on regulatory decision-making in the future. And also, we will see some more presentations throughout today's track. And so I hope this gave you some flavor how pharmacometrics can support the post-approval stage, and we will see some other examples later on. And I will take any questions at this point if we have.
Thank you very much, Oksana Jones. That was a really nice summary of a variety of ways that models can help with post-approval requirements. We do have a couple moment for a few questions. One I had for you as a moderator, I kind of get to take advantage of asking my question first. A lot of drug sponsors have a focus on the submission and the approval of their drug product. When you're building these models to assist with sponsors, is there a way to improve model and the success of those models and utility of them? Is it better to start earlier in the development of the drug development life cycle for that product? Or is it better to wait until you have the conclusion, the clinical phases where you have more data that you can kind of pull together in a single analysis?
Yeah, that's a very good question. And I think it's a very good question as from a consulting perspective, since we usually get into the game when it comes to the approval, right? And when we are there. So I would say to answer that question is the earlier you can start and gain your knowledge and know the knowledge gaps which you have to fill and by utilization of the models, the better it is. So definitely, I would say along the way, and then you can bridge those knowledge gaps also by informing some of the drug development decisions of like, where are gaps and how can we fill them? How can we make the models more robust and better to answer then later on those questions in post-approval.
And then kind of follow up, you've showed a wide variety of different types of models. In your work with various drug sponsors for post-approval needs, have you found that, there's kind of a one catch-all model that you can build for them, or is it really kind of tailoring the type of model for the data available and the questions being asked?
Yeah, that's a very good question as well. It is often also tailored. I mean, you might have like specific questions, right? And you might pull in certain portions of your studies and focus on certain portions. Let's say when you think about the pediatrics, your focus will be more on maturations, on body weight relationships and things like that. So keeping that in mind, you might spend some more time quantifying that really well and knowing your gaps and what can be supported. So yeah.
Excellent. I was going to say, I don't see any questions posted or any hands raised for audio questions. I think we're close enough that we can kind of move on to the next session and kind of save some time as we go. As Lindsay posted in the group chat, please make sure that you leave this session and then join the next session, which is the FDA's perspective on the PBPK analysis of biopharmaceutical applications presented by Banu Zolnik. Thank you very much. I'll see you at the next session, everybody.